This video is about the diffusion of molecules into bacterial cells. Diffusion is the net movement of molecules or atoms from a region of high concentration to a region of low concentration as a result of random motion of the molecules or atoms. There are three types of diffusion: simple, facilitated, and active diffusion. Active diffusion is a diffusion in which the molecules move against the concentration gradient, meaning they move from a region of low concentration to a region of high concentration. Factors affecting the diffusion rate is the number of protein channels, the temperature, and the size of the molecules. The first diffusion that we're going to look at is called simple diffusion. It requires a concentration gradient, and the molecule should be very small and lipid soluble. It's passive and doesn't require any carrier proteins. The molecule that we are going to use in this example is oxygen molecules. This is the phospholipid bilayer of a bacterial cell. For simple diffusion, I've decided to exclude the carrier proteins because they're not needed. The purple circles in this example should be regarded as oxygen molecules, and the number of oxygen molecules on the outside of the cell are greater than those that are inside the cell, meaning there's a concentration gradient. And because oxygen is a very small molecule and is lipid soluble, they're just going to diffuse right through the phospholipid bilayer. Here it shows that diffusion did take place because the number of oxygen molecules on the outside is less compared to the number of molecules in the inside. The second diffusion is called facilitated diffusion. It also requires a concentration gradient. It doesn't require energy, it's passive. It requires carrier proteins, and it's for large molecules or charged molecules such as glucose. These are the carrier proteins which are required for facilitated diffusion. As you can even see, the circles here are larger, showing that these are large molecules. Let's just call them glucose for this example. Here, the number of the glucose molecules on the outside of the cell is greater than the number of those that are inside. These glucose molecules are going to diffuse through the carrier protein into the inside of the cell. Here it shows that some of the glucose molecules did indeed diffuse through the carrier proteins to the inside of the cell. The last one is called active diffusion. It requires a concentration gradients, it requires energy in the form of ATP, carrier proteins, and is also for large and charged molecules. In this example, we'll be using urea as our molecule. Urea is a waste product, so it will be removed from the cell. These are the carrier proteins which is required for the active diffusion. Here it shows that there is a concentration gradient, but in active diffusion, the molecules move against the concentration gradient, meaning they move from the region where there's low concentration to a region of higher concentration, and it requires energy in the form of ATP. But as soon as the ATP binds to the carrier protein, a phosphate group is released from the ATP, forming an ADP. And the energy that is created when the bond between the ADP and the phosphate group creates energy, which is going to be used to get urea molecules from the inside of the cell to the carrier protein and to the outside. Here the molecule binds to the carrier protein and is pushed to the outside by the energy which was created when the phosphate group was removed from the ATP. Thank you. This video is about the diffusion of molecules into bacterial cells.